Okay, so everybody has finished streaking your starter plates. And y'all washed your hands without me even telling you. That was awesome. We are working with bacteria. It's a good idea. We're going to talk a little bit now about the plasmid that we are going to try to give to your bacteria tomorrow. Right now, we're just growing some bacteria because you need bacteria that are actively growing in order for them to take in DNA from their environment. But we're going to talk a little bit about the plasmid that we're going to give them. These next few slides are in your note packet. You can also have the lab document open in front of you because you probably saw there's a question that asks you to explain the genes on the plasmid. But if you want to just jot some notes in your note packet as we talk through this and then summarize it in the pre-lab, that's up to you. If you are following along in your note packet, you should find a slide that looks like this. It also has a diagram of the pGLO plasmid itself. So in this lab, we are attempting bacterial transformation. And if you remember from last week, transformation is when bacteria take in genetic information from their environment. So what we're going to do tomorrow is we're going to take some of the bacteria that are growing on those plates and put them into a liquid where we will add a plasmid that we are hoping some of those bacteria will take in. And then they'll start using the genes on that plasmid and we will see some of the new traits. The plasmid that we're giving them is called a pGLO plasmid. It is a recombinant plasmid that was engineered in a laboratory to have a particular set of genes that we want these bacteria to get. If the bacteria successfully take in this plasmid, they will copy the plasmid just like they copy all the rest of their DNA every time they reproduce. And so all of the offspring of any bacteria that takes in this plasmid will also contain this plasmid that has all of these genes. So you can see on this slide a map of the genes that are located on the pGLO plasmid. This part over here, the ORI, stands for origin. So that's the origin of replication. So when this plasmid reproduces, when it replicates itself before binary fission, that's where DNA replication begins. That's where the double helix starts to come apart and the new strands start to be built. There are 3 other genes located on this plasmid. There's one called ERA-C, there's one called GFP, and there's one that's BLA. And we'll talk about what each of those genes does. The ERA-C gene codes for a protein that acts as a regulator for the arabinose operon. Arabinose is a sugar, and this type of, these genes, ERA-A, ERA-B, and ERA-D that you see along this operon are genes that make enzymes to help the bacteria break down the sugar arabinose. So this ERA-C codes for a regulatory protein. So this gene is kind of like the one that we labeled I in your operon notes last week. It's not part of the operon itself, but it codes for a regulatory protein that you can see is located at the beginning of the arabinose operon. And so that protein acts to help regulate when these genes to digest arabinose are turned on and when they're not. The way that control works looks like this. So you can see in that previous slide that ERA-C protein is kind of in a vertical, it was kind of in this kind of conformation. And when it's like that, it's really hard for RNA polymerase to bind. And so those arabinose genes are not transcribed and translated. But when arabinose is present, so this is that sugar, when this sugar is in the environment of these bacteria, that sugar, arabinose, binds to that era C protein and changes its conformation. It changes the shape of that protein in such a way that now RNA polymerase can bind, and these genes begin to be transcribed and translated. So thinking about what we talked about last week, would you call this operon inducible or repressible? And is this an example of positive or negative control? Turn and talk for just a minute. So this is an inducible operon. It's able to be turned on when arabinose is present. This is also actually an example of positive control because this era C protein speeds up the transcription of these genes. It helps RNA polymerase to bind and start transcribing and translating these genes. Now, what we did in the pGLO plasmid, we meaning the scientists who genetically engineered this plasmid, we kept the control mechanism. We kept this part, the fact that arabinose can bond to the era C, which helps polymerase bind and transcribe, but we cut out these genes. We don't actually really care if our bacteria can digest arabinose, but we want to use that switch. So when there's arabinose there, arabinose acts as like an on switch to turn on a gene that we put there instead. So we took out all of those, the genes for the arabinose digesting enzymes, and instead we put in a gene called GFP. This stands for green fluorescent protein. This is a gene that we got from jellyfish that glow under ultraviolet light. And so if bacteria express this gene, if arabinose is present and turns on this operon so that this GFP gene gets transcribed and translated, these bacteria will produce a protein that will make them glow under ultraviolet light. That's how we'll be able to tell whether these bacteria have successfully taken in this plasmid and whether they have this gene turned on and are actually transcribing and translating it so that it works. In order to select just the bacteria, really the only bacteria that we want to grow are the ones that successfully take in that plasmid. And so in order to isolate those and kind of just grow the ones that we want, there's another gene on here. This gene right here, it's called the beta-lactamase gene, produces a protein that makes the bacteria resistant to an antibiotic called ampicillin. So what we'll do is we will try and get our bacteria to take in this plasmid, and then we will grow some of those bacteria on agar that contains ampicillin. If they successfully took in this plasmid, their cells should be reading that plasmid and producing this beta-lactamase protein that'll make them resistant, so they'll be able to grow anyway. But if they did not take in the plasmid, they will not have this resistance and they will not be able to grow. So the only bacteria that should be able to grow with ampicillin in the environment are ones that have successfully taken in this plasmid. Does that kind of make sense? So we can use this to kind of select. It'll kill off any bacteria that did not receive the plasmid, and the only ones that'll be able to glow are those, or that will be able to grow on those plates are the ones that did take in this pGLO plasmid. Questions about that at all? All right, I think, yep. So that's the genes that we have on the pGLO plasmid that we're going to try and give to your bacteria tomorrow. Before tomorrow, please complete those couple pre-lab questions, and you can answer the part about what the genes on the pGLO plasmid do. It's also really important, please read the procedure before you get here tomorrow, maybe twice. This is a really specific procedure where things have to be moved from one temperature to another for an exact amount of time. And if we don't do this right, the bacteria won't take in the plasmid and this transformation won't work. So please read it before you get here so you have some idea of what's coming ahead, because it is fairly time sensitive. And the operant homework questions are actually due tonight, not Wednesday. Sorry. Old deadline. Any questions at all? All right, that's it for today. Have a great afternoon. Those of you at home, we'll see you tomorrow for some transformation. Yeah.
Did you find a way to return that?
Oh, yeah.
Google form? I was gonna ask you about the last question.